Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, C-suite veteran Peter Traber, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on this week's edition of Surfing the NASH Tsunami.
For everyone with an interest in NASH or more broadly, fatty liver disease, surf's up because episode 28 of Surfing the NASH Tsunami starts now. I'm back in New Hope, Pennsylvania, self-quarantining after my week in Corpus Christi. For those who are not in the U.S., the way we deal with having all these COVID cases is that if you come from parts of the country with high COVID rates and you come back to a part with a lower COVID rate, you are told to keep to yourself for 10 days or 2 weeks, and that's what I'm in the middle of doing. Now on to more pertinent subjects. One piece of feedback we've gotten recently is that we're overdue for a Wild Times in NASH-Ville style drug development episode. Episodes 5 and 7, we talked about drug development back in, May that was. And Naeem did a really great job last week discussing some of the upcoming clinical trials we anticipate in the fall. But the week's format didn't allow us really to dive into those implications for a larger market. Today we're going to do that. We're going to do Wild Times in NASH-Ville 3, and Stephen will lead the regular surfers, Louise, Donna, and me, through a discussion on the topic. Here at Surfing NASH, we're delighted that listenership continues to grow. I mentioned last week that episode 25 had the biggest first-day uptake of anything except ILC and the Intercept CRL. Well, 26 was bigger, and we haven't seen the full week on 27 yet, but it looks as if that's going to be bigger as well. So thank you for your continued enthusiasm. Two more things before we get started. As I mentioned last week, we are going to start making decisions about exactly what our digital AASLD coverage looks like this week and next. Please, if you have any input, anything you want to see or hear, share with us on the discussion group or send a note through the email address. I've already gotten a couple of comments. I've had conversations with people. I have 2 more scheduled, and that will help us get you the coverage you actually want. Next week, we'll talk a little bit about what we're going to do. Finally, for music, we're going to head back to Jeff Ramsdale and the British Liver Trust. We have special reason to honor the British Liver Trust this week. On Monday, they kicked off a major campaign called Sound the Alarm About Liver Disease, which will be the subject of a future episode. And by future, I mean maybe next week, maybe the week after that. So on to our podcast. We're going to do something a little different today. We're going to start with whatever kind of highlight anybody chooses, personal or professional. Brave one, go first.
I am happy to be celebrating the 26th anniversary of my liver transplant today.
Hear, hear.
And I celebrated it with both a bike and a bar. That's with 2 R's, a ballet-inspired workout this morning. And I'm just feeling so grateful and privileged to have made this milestone. And so thankful, so thank you to all the surgeons, all the nurses, all the medication innovators for putting all the pieces together. I want to thank my insurance company for covering things. Don't want to leave them out. And just thank you to my family as well for standing with me and by me throughout all these years. And I look forward to what I can do to make sure that other liver patients, first of all, don't need a transplant, but if they do, that they are able to get a transplant and live life even more healthfully and successfully as I have.
Donna, on my part, and I speak for all of us, a heartfelt God bless to everybody who helped you get to this day and gave you the gift of life, because we then all get the gift of you and everything that you do for the community and to brighten our lives, and it's just fantastic. So 26 more, okay? I know it's going to take a lot of doing. 26 more.
I'll go. Donna, you shame the angels.
I love that. I'm writing that one down. Thank you.
So how can you stand up to that? I can say I've been married 26 years. This is my 26th year of being married. I'm 51, which means I've been answering to my wife longer than I was single. So for what that's worth, I'm still standing here, maybe for the better. I'm sure it's for the better. For me, it's just really my son contracted COVID at what we call COVID Station. It used to be called College Station. It's the town where Texas A&M is. But it's affectionately earned the reputation of COVID Station. So he's better. His toughest part of the whole deal was quarantining in his little bitty teeny weeny room for 14 days and not being able to exercise, people had to kind of slide food around the corner of the door, but he made it through. There is an interesting cocktail that I gave him that I wonder if it's being studied. I gave a double dose Zyrtec and high dose Pepsid twice a day to block the histamines, and whether he was getting better on his own or not, it seemed like that helped him. So either way, he's back to the grind, back in his coursework and doing well. So it's good to have him back and be done with it. So hopefully everybody else is doing well in addition on the podcast and those listening. But if you're not, Godspeed, and I hope you a speedy recovery.
Stephen, glad to hear about your son. Louise, what you got?
Well, hats off to both Donna and Stephen, and I'm glad everybody's well. And I can't really compete with any of that, so I'm just going to stick with the open 2 clinics in Harley Street that were fully booked, which was excellent news. So hopefully get some more and start opening up more of our clinics. Even though we're starting to lock down a little bit more, it's still encouraging and people are out there wanting their diagnostics. So that's mine.
I'm just glad to be back in Pennsylvania. We live in a small condo on the river. And as I'm doing this podcast from home, I'm looking out at the beginning of fall foliage and the trees on the other side. And we've got ducks and geese and swans and osprey and all kinds of assorted birds swimming in the river. And it's a time of year that you really appreciate the diversity of life in its broadest sense. I feel lucky to be in this place. I feel lucky to spend time with my granddaughter last week. And these are all good things. And let's just leave it at that. Moving ahead. Today's format is simple, which is Stephen's going to take some time right now to talk about the recent 89 Bio press release dated September 14th. We'll have a link to the press release itself in the episode write-up, and issues in general related to drug development for 89 Bio, maybe Caro, FGF21s in general. And then we'll take some questions, and then we'll branch out beyond FGF21 issues to think about other drug classes, other agents in development, and what this means for the field in general. So, Stephen, you're up.
Well, first of all, it's great to be back after being off last week. I hope you all missed me terribly. There was a special place in my heart that was empty, so I'm glad to be back. So what I want to spend a little time talking about today is the FGF21 class of drugs for the treatment of NASH. And the reason for that is because as Roger mentioned, there's been a recent press release by another company who is developing this mechanism of action for the treatment of NASH. So this actually makes the third company to present initial data on an FGF21 agonist. We have Bristol-Myers Squibb leading off with pegbelfermin, which reported their results a couple years ago on a phase 2A proof of concept non-invasive test evaluation. That showed positive results and led to the initiation of 2 large phase 2B paired liver biopsy studies, which are currently underway and should be finishing up soon. We're anxious to see results probably in the first part of 2021. So 89 Bio is the company that I want to mention today. But before I do, I want to remind the audience about FGF21. It's an endogenous metabolic hormone that regulates energy expenditure and glucose and lipid metabolism. And very typically, it has a short half-life, less than 2 hours for a native FGF21 molecule. And the way our understanding of the biology of this hormone working is that it basically works through binding to FGF receptors 1C, 2C, and 3C, and really it doesn't have any activity against R4, which is really the way that aldafermin or NGM282, which is an FGF19 analog, works. So in binding to FGF receptors 1, 2, and 3, you downregulate de novo lipogenesis in the liver, which counts for about 30 to 40% of all the fat in patients with NAFLD. And it also works on adipocytes and skeletal muscle to inhibit lipolysis and increase glucose uptake. And by doing that, you prevent or reduce free fatty acid flux into the liver, which is the primary mechanism by which patients with NAFLD get fat in their liver. So you're blocking essentially 2 out of the 3 main mechanisms by which patients with NAFLD get fat in their liver. By default, you would think that the drug would work very well on defatting the liver in patients with NAFLD. And we saw other results from Akero that highlighted that fact very elegantly with the significant drops in MRI PDFF that have been reported. So focusing back on 89bio, their FGF19 agonist is termed Bio 89-100, and it is an engineered FGF21 that allows for a much longer half-life of approximately 55 to 100 hours as opposed to the native FGF21 half-life of less than 2 hours. They get there by adding a glycopeg moiety onto it, which increases the half-life. They also mutate 2 positions, 173 and 176, and the glycopeg is attached at position 173. And so doing, they maintain similar binding to native FGF21, but they prolong the half-life. So the interesting concept about the engineering of all of these FGF21s, whether it's BMS or Akero or 89bio or others that are in development, is that they're all a little bit different, which if their chemical scaffolding is a little bit different, potentially their efficacy and their AE profile have the potential to be different as well. So moving forward, the company reported top-line results from their phase 1A, 1B, or it's kind of an extended 1A trial where they basically looked at safety, PK, and relative changes in liver fat, as well as serum lipids, liver and metabolic markers. They ultimately enrolled about 80 patients. They had 81 total enrollment, 71 completed the study. And the treatment duration was 12 weeks. So there was an MRI at baseline, an MRI at week 12. And the study population was a generally mild NASH population. You did not have to have liver biopsy necessarily to get in. You could have what we call phenotypic NASH, which was based on liver enzymes and a FibroScan criteria. And when you look at the baseline characteristics of the population, you had an ALT mean of about 42, AST mean of 31, MRI PDFF 21, so significant amount of fat, but mild to moderate elevations in ALT and AST, more probably on the lower end relative to F2, F3 studies that we have seen in NASH, but about 40% were diabetic across the population. So I would define this as kind of a mild NASH population, if you will. F1s, probably some F0 NASH in there as well. Top-line results were impressive. They had significant drops in absolute liver fat content. I will say this, I didn't mention this at the beginning. There were 6 different dosing arms of the study, 3 milligram, 9 milligram, 18 milligram, 27 milligram, dosed once a week. And then they had 2 arms that were dosed every 2 weeks, an 18 milligram arm and a 36 milligram arm, and then a placebo control. So absolute liver fat drops across the board were about 7.5% up to 13.5%. That was with the 27 milligram once weekly. The Q2 week dosing had absolute drops in liver fat of 9% and 9.7%. Focusing on some data that I find equally important, if not more important, in beginning to look at how this might translate into histopathology change, I look at relative liver fat reduction, and particularly the proportion of subjects with more than 30% relative liver fat reduction, because that's where we have data in other mechanisms linking to histopathologic change. When we look at the Q weekly dosing from 3 milligram up to 27 milligram, we see about 60% up to 86% of people achieving that magic 30% relative fat reduction. And it was 69% and 88% in the every 2 week dosing with 18 and 36 milligrams respectively. The other bit of impressive news with this is about 43% of subjects actually normalized their liver fat content over the 12-week period. So how did that correlate with reductions in ALT? Remember back to baseline, ALT mean was around 40, so it wasn't significantly elevated. But having said that, if we focus on the 27 milligram once weekly dose, there was a 30 unit per liter absolute change across the board with that dose. And in the Q2 week dose at the higher end, 36 milligram, there was a 22 unit per liter decrease. So ultimately, at least with the higher doses, we were seeing pretty profound drops in ALT. So maybe I'll just stop there and see if you guys have any questions before I continue to highlight some of the results.
It's not that I don't really have a question. I thought that was a great summary of what looks to be quite a nice drug. I think it had minimal side effects as well. My only comment about a lot of these drugs seems to be that they're subcutaneous injections, and some patients don't respond well to that idea. A lot of them get over it, but how do you think that might actually complicate pathways going forward if it does get more of an uptake in phase 3 and bypasses all of that?
I mean, that's a great point. I mean, patients, I guess I find that the more severe the patient perceives their disease to be, the more willing they are to go outside the bounds of a normal oral tablet to get the benefit, right? So the classic is the juice worth the squeeze phenomenon. So I do think if you're an injectable that you have a little bit of a higher bar than if you're oral. And if you're an infusion, you have a higher bar than an injectable and an oral tablet or pill. So I think those are important points. So not only when I say higher bar am I looking at histopathologic change, but I begin to broaden the aperture a bit to say, what are you doing for overall metabolic health? You know, are you changing insulin resistance, dropping hemoglobin A1C? Are you doing anything to lipids? What about weight? I'll just jump right in and just tackle that, because that was also described in the press release that there were significant reductions in triglycerides. If you look at the percentage change from baseline at week 13, we see changes from 18 to 28% in triglyceride levels. They did a separate analysis in a subgroup that had baseline triglycerides more than 200 and showed that there was a 33 to 49% decrease from baseline in that population. Broadening out to the other lipid moieties, there were changes in non-HDL, changes in LDL cholesterol, as well as improvements in HDL cholesterol. So it was kind of a good profile, at least on the lipid changes that we're seeing. As far as glycemic control, there were significant changes in adiponectin, and we know that that is a hormone that goes up as you improve insulin sensitivity at the adipocyte level. So that's kind of just a marker saying that, in fact, that is happening with this drug. But there were also positive movement on insulin and minor changes, non-significant changes on hemoglobin A1C. Again, hemoglobin A1C is a marker of glucose levels averaged out over several months. And remember, this trial was only 12 weeks. So that's a caveat where maybe the hemoglobin A1C wasn't moving because there wasn't enough time to see a change, or it's not moving because this drug isn't really impacting glucose that much. I think it's too early to make that call. But there were some positive changes in insulin. And as far as safety goes, at least what was reported, it appeared to be well tolerated with very few treatment-emergent adverse events, certainly none leading to death. There were 2 patients that stopped medication due to an adverse event, and there were 2 serious adverse events noted. Just backing up to the treatment discontinuations, one was due to a skin rash and one was due to hyperglycemia that was not deemed to be drug-related. And then on the serious adverse event side, the 2 cases that were there were felt to be attributed to COVID-19 and not drug-related. As far as what they saw from an adverse event profile, it's very similar to what we see with any of these hormone agonists that work on FGF receptors, whether it's 19 or 21. They tend to be GI related. The GI adverse events here were similar to placebo. About 15% had increase in appetite, 13% had diarrhea, and 11% had headache. And the only treatment-related adverse event reported in more than 10% of the pooled drug-related group were mild increases in appetite. So, just putting this in perspective, which I think is important with the other FGF21 compounds, this was a 12-week treatment trial. The other more recent FGF21 agonist that has been reported in a press release is efruxifermin. Try pronouncing that. And that was with 16 weeks of treatment, 3 different doses with relative reductions in liver fat of around 60 to 70%. And then prior to that, I mentioned early on in the podcast pegbelfermin, which is Bristol-Myers Squibb's FGF21, and in its early phase 2A study, which was a non-invasive trial, treated for 16 weeks, so same duration as efruxifermin, we saw changes in liver fat of 20 to 32%. So stacks up better than pegbelfermin and about the same on liver fat reduction as efruxifermin. So maybe I'll stop there and listen to any comments or questions so far.
So this is Donna. I was thinking back to your comment several episodes ago about, combining the best of various mechanisms. And so, is this an example or a candidate of having something that's highly effective in the fat reduction and pairing it with something else that might be highly effective in fibrotic reduction to have sort of the best of and the best chance of a holistic treatment for patients?
Yeah, that's exactly right. I think what you would ideally want is have a drug that works very efficiently and effectively with a low AE profile that's very safe and well tolerated, that can defat the liver. And if you can defat the liver through inhibiting de novo lipogenesis or by inhibiting lipolysis and increasing glucose uptake, that's terrific. And if you can add to that something that could augment the effects of that, then I think you've got a wonderful combination. When I think of 89bio, I reflect back on the fact that this was a small proof of concept, early phase, non-invasive trial that gives us a lot of excitement relative to the changes we're seeing in MRI PDFF, based on the fact that when we look at more recent data from other sponsors with different mechanisms of action or even the same mechanism of action, we see that significant drops in liver fat content do manifest itself in the form of increased NASH resolution histopathologically and improvement in fibrosis. Maybe more importantly, we're moving past the 30% relative liver fat reduction, and now that we have drugs that are pushing the needle on 50% liver fat reduction and even 70% liver fat reduction, what happens to those people from a histopathologic perspective? And Rohit Loomba presented the resmetirom data at the digital ILC and really kind of began to highlight some of this newer data coming out, looking at a even larger magnitude of effect change in liver fat content, showing that if you got to the 50% mark, 60% of people resolved NASH, and of those, 60% had improvement in fibrosis, and overall, 30% of patients had improvement in fibrosis, hitting that number. That is encouraging, and it begins to allow us to think that defatting the liver allows the liver to do what it needs to do, which is begin the healing process, right? It's the old adage of cure the C, suppress the B, quit drinking alcohol, and treat the autoimmune. If you do those things, the liver will get better on its own. And there's been a lot of debate over the years about, if you just defat the liver, is that all that's necessary? I think we're beginning to see that if it's not all that's necessary, it is a big component of it. So where you have drugs that are able to do that, I think that's highly encouraging. I think for 89bio, they need to show us They need to generate some histopathology data, and I think that's hopefully in the works for them to do that in future studies. But to your point, if we can take an oral antifibrotic or even an injectable antifibrotic, and we can combine it with a drug that moves the needle on metabolic profiling, defatting the liver, improving insulin resistance, improving lipid profiles, those are really the components I want to see. So let me just summarize. If I can have a drug that defats the liver, improves insulin resistance / glycemic control, improves lipid profiles, and can plus or minus augment weight loss and work on fibrosis, I think those 5 components are really what we need to help our patients from a long-term outcome perspective as it relates to liver-related outcome and as it relates to cardiometabolic outcomes or CV outcomes. The way I see it, that's critical. And along the way, something that we leave off often is how a patient feels as well. Because remember, the FDA approves claims, not drugs. So if we're able to show a patient has a change in how they feel, function, or survive, that's critical to getting approval. We seldom talk about the feels part, but we now know through amazing work that Zobair Onasi has done at Inova Fairfax and others that when we measure patient-reported outcomes from baseline, that patients do feel worse with NASH, and that if we can impact that change positively, that's also important and something that we should be measuring along the way.
Stephen, first of all, thanks for a brilliant 20-minute education and where we're headed with FGF21s. I've been trying to scribble notes, which is hard for me to do while running the podcast. I know the idea that Bristol-Myers Squibb compound is belfirmin says pegylated. The native half-life of the molecule and how long it's got to be in the body says pegylated, and you described some kind of glycopep process for the 89 bio medication. What's the meaning of the different forms of pegylation in terms of efficacy or half-life?
The pegylation process just allows for it to not be broken down as rapidly in the bloodstream, right? So it hangs around longer. You don't have to pegylate to get that, right? There are other, FGF21s in development that, that work to extend half-life by alternative mechanism. Caro has one, right, that it's not pegylated, but they have certain proprietary point mutations that basically allow for increased stability of the drug and longer half-life as well. And Caro's drug is quoted as having a half-life of 3 to 4 days. So very similar to that 55 to 100 hour half-life that 89 Bio reports.
Stephen, when we're talking there about defatting the liver being a hugely positive and in some of these drugs very rapid, given that steatosis and liver fat is linked with the development of type 2 diabetes, obviously cardiovascular disease, the hypertension, the dyslipidemia, is there a potential role for these medications outside of the development of NASH, so earlier and NAFLD in the prevention? We talk a lot about lifestyle and altering and locating earlier in the timeline, but if defatting the liver reduces the risk of type 2 diabetes development or cardiovascular disease, is that where these medications could also be targeted? Because, of course, non-communicable diseases, massive mortality rate around the world. So finding it earlier may well become more of an option to prevent those comorbid conditions, really.
I mean, I think that when you defat the liver, we talked about this on many podcasts ago, that the liver is the canary in the coal mine for metabolic sensing. So where you're able to defat the liver, whether that's through lifestyle change or pharmacotherapy change, I think, it bodes well for metabolic health. And if that's the prevention of diabetes, if it's the reduction of cardiovascular disease, then potentially there's a role. The problem with all of this is that it has to be proven. So, a lot of these companies have chosen to take the NASH route as the way that they would get approval for their drug. Semaglutide, for instance, the GLP-1 agonist, chose other pathways first, right? Because the GLP-1 agonist, there are no GLP-1 receptors in the liver, but they work within the gut to lead to weight loss, and in so doing have profound effects on hemoglobin A1C and glycemic control. So, Novo Nordisk took the route of going after a diabetes indication. They're also studying an obesity indication, and then they got into NASH. So I think, if you look at other drugs like resmetirom and even 89 Bio, they're being looked at for their hyperlipidemic, their anti-lipidemic effect, their effects on lipid metabolism as a potential another mechanism for approval, potentially. But I think, you have to start somewhere, and then you have to show data that supports your claim. And it's just a matter of priorities within the company, I think. I don't know if that answered your question, Louise.
Yeah, it does, and I think it's about, as you say, about the evidence. I just wonder in the days now of COVID, we know that a lot of these patients are the highest risk of outcomes, and there will be a future pandemic. We've already seen SARS, MERS, H1N1, whether or not it remains now either ethically or politically appropriate to let people develop type 2 diabetes when we can do a large prevention strategy and we can target that, or if it's acceptable to develop cardiovascular disease. I don't know now whether or not we can sit here and say we're going to only treat the end stage, but we know we need the evidence. So I just wonder where we sit ethically and politically in allowing people to develop these diseases when we know that they're so intrinsically connected. That was just a thought I was having when I was listening to all of that and having seen a lot of the press over the last few weeks.
It's a great comment, and I think a real consideration. I'm hoping in a couple of weeks to put together an episode on COVID and the liver, where one of the things we'll get into is exactly that. I think not what does COVID mean for patients with pre-existing liver disease, but what does pre-existing liver disease do that has an impact on COVID, and as a result, does that make it more important to think about treating earlier? I agree with you. I think we're going to hear a lot more on those kinds of issues, not just on this podcast, but in the world as we go forward. Stephen, I had a commercial question. As Donna pointed out, and as we've all noted, the ideal strategy, if I was only focusing on the disease, would be to get two really great agents that worked well in combination and put them together. The reality of commercial drug development is that that's likely to happen if one company has both agents or buys a company that has a second agent, or two companies link up. I'm wondering if you're aware whether there's anything else in the pipeline of any of the FGF21 companies that would lend itself to that kind of combination. And let's extend it. You also mentioned aldafermin and resmetirom and maybe a couple of others. Is there an obvious place right now that you would look and say this company has what it takes to put together the right combination and be studying it now or quickly?
Well, you're absolutely right. Kind of the drug development landscape in the field of NASH now has been, I would say for the most part, and I think Gilead is the exception to this rule, they've been focused on their one asset and their one mechanism of action and kind of getting it across the finish line before risking combination with another agent. And I say risking because, there are concerns that if you combine your drug with another drug prior to approval, that maybe you run across an adverse event or some other safety issue that would potentially derail your monotherapy development. Whether that's theoretical or real, it's certainly a concern. If you have drugs in-house, like Gilead had several different mechanisms of action. You saw from the digital ILC the Atlas trial, which was a 392 patient paired liver biopsy study with multiple different combinations of 3 different mechanisms of action. You can begin to do that kind of combo work. Now, to my knowledge, when I think of the FGF21s, I am not aware of that kind of combination work being done in-house or for partnering, but I may just not be in the know on that. I will say this: there are companies out there in the NASH field that do have exciting assets under one roof where combinations can begin to be had. And I think those are in the works. And so that's exciting to see. But what I wish would happen is that drugs that show positive impact on histopathology and that also have some kind of movement in a positive direction on some of the comorbidities of NASH, whether it's lipids, glycemic control, insulin, weight, that those would begin to be combined with other drugs that are moving the needle. You know, we've seen combination therapies between companies. You see it with Novartis and I think with Allergan or now AbbVie, and you see it with Gilead and Novo Nordisk. You know, ideally, to me, the perfect drug combination is taking two drugs that work well on their own and either would have a positive synergistic effect on histopathology or they would have a positive synergistic effect on lipids, glycemic control, weight, or something like that. Or if there's an adverse event of one, that the other drug would mitigate the adverse event. So like FXR is a great example. It may still be the first drug approved for the treatment of NASH. We're all hopeful that obeticholic acid will be that, but it has a liability in the form of LDL, but it does move the needle on fibrosis and to some extent on the components of NASH as well. So, you know, we've seen positive impacts on the thyroid hormone receptor beta class in resmetirom, moving the needle on NASH resolution, maybe at least in the phase 2b, more so than fibrosis. And it significantly lowers LDL cholesterol. So it's oral. It's once a day, so is obeticholic acid. So if you put the two together, do you augment the histopathologic benefit while also mitigating the LDL issue? And you know, those are the things that I like to think about when I think of drugs that would optimally work well together. Now, look, Scott Friedman is an amazing man. He is a wonderful mentor to me and to many others, and he has no issue sharing his knowledge, and for that I'm eternally grateful. He's taught me along the way a lot about what can happen from a transcriptomics world, what can happen from a in vitro work, from a cell culture model to an animal model to, a genetic transcript model where you put these drugs together and you see what genes are moved in a positive way. And are they the genes you want to see move relative to this disease state? And then you study that combination. That's another way to get at it versus waiting until you have two drugs that clearly show a histopathologic benefit. But whether you do it that route or you go the way that I mentioned, it will be important that these combinations happen. And I think we're going to continue to see more of this as we move forward. And I think that's the future.
I think we all agree and can't wait to get there. Louise, Donna, questions?
I'm always so excited to learn, and I've learned so much today. I'm absorbing a lot and putting these pieces together. That has been, my great privilege being involved as a patient and patient advocate in this field over the years, whether I'm in, Paris or Utah or now on Zoom, to be able to hear directly from folks like Dr. Stephen Harrison, not only, the scientific details of each mechanism and how they potentially fit together, but the broader implications for patients. So if, people ask how Global Liver Institute developed its policy, it is hopefully reflective of the time that I get to spend listening to conversations like this.
Seconded. Louise?
I echo Donna's sentiments there. It's been interesting to listen to more of the science. That's not my strength, but looking at how these medications can be utilised together and how people can take them, maybe as Stephen was suggesting, in combination, DFAT. It becomes more holistic to patients. It becomes more intricate to each individual. You can design pathways for each specific sort of patient. So it becomes more individualised. And I think the more options we have, the better we can get If they pass the tests, if they go through all of the clinical trials and are positive in the end, then the future's looking bright for more and more specific individualised care programmes for patients with all of these multi-morbid conditions. And I think that's got to be exciting. Obviously, we need the evidence, but it's certainly a good start.
Amen. Stephen, I had a conversation earlier today with one of our listeners, a treating physician, who said one of the great things about this podcast is I only get to learn what KOLs are really thinking if I body block somebody like Stephen Harrison on the way out of a meeting. So I think that individual, for having to save body blocking and getting to hear this, and the rest of us are really privileged, and thank you for that. Do you have a final thought or takeaway on this subject you want to leave people with, or do you feel you've covered all of the landscape already?
I think we've done enough for today. We'll have many more times to discuss how this fits into the bigger picture, but I think that there's a lot to digest for today, for sure.
So why don't we just go on to our close? And biggest surprise, most important thing you learned, or the one thing you're really going to walk away thinking about from today's conversation?
I'll jump in on this one. I suppose it was Stephen's 5 needs from the treatment, so the defatting, the improving insulin resistance, lipid profiles, with or without weight loss, and working on fibrosis. And I think if we can achieve anything that has those 5, and we put the ability of a patient to determine their outcome, then we've got a powerful set of treatments that could be there. So that's what I'll take home from today.
I agree with that. That stood out to me for its clarity. I also wonder how many years it would take us to reach that and what the interim steps look like, and specifically the regulatory process for getting something before that. I fear that we let the perfect be the enemy of the good, and that as there's an urgency today for patients to have a treatment that gives them even some modest effect, that while we keep our goals on what the ideal is and what we are ultimately working for, we don't let that foreclose options to have some incremental steps towards that ideal goal.
Donna, I was thinking the same thing about how do you get there and how do you get agents approved, particularly because the other thing that I heard was when we start talking about going past 30% PDFF reductions to 50 and 70% PDFF reductions, it feels to me like we're talking about generations of drug development out. But the faster we can get started on actually educating the population about how to use drugs that are good, if not perfect, or good, if not great, the more we'll spur development, and the more we'll spur everybody to think differently and more affirmatively about what they can do to treat NASH specifically, and NASH in the constellation of all the metabolic diseases. So I'm with you. I wound up thinking about how do we get started, and that I think is a regulatory issue more than anything else. So I agree. Stephen?
You know, I kind of used up all my words, I think, during the podcast, but I think, you know, ultimately we're seeing very positive impact from targeting FGF21, and I think that this is very good because we've seen positive movement this year from the work being done with GLP-1 agonist, for the work being done with thyroid hormone receptor beta agonist, the pan-PPAR data that's coming out, the FGF19 data that's coming out. And so there are many other mechanisms that are being studied. And while we've had some setbacks early on with some of the trials, some of the combination trial data, some of the monotherapy data, the picture is becoming clearer as it relates to the pathogenic pathways that could be targeted both individually and in a combination format, where we're able to get positive data. And I think it's a growing pain, right, where we get all excited about NASH and everybody jumps in to drug development, And then we have some setbacks with drugs that don't work. They don't quite have enough, efficacy to change histopathology, and people begin to shy away from the field. And then we come back stronger as a result of it with newer therapies, newer agents, a better understanding of the pathology of the disease. That's where we're at now. We've just climbed a mountain, and now we're coasting again. We're generating some positive data. And for the listeners out there that have the disease, that study this disease, that are working in the field of drug development, it's exciting times again. And I think we'll see this momentum. We're going to ride this tsunami wave, if you will, all the way into 2021. And it's good news for our patients. And for those people like Donna and Louise and Wayne and others that are huge advocates for patients with this disease, it's great to be able to deliver this news. We're not there yet. We have a great amount of work still to be done on disease state awareness and getting the non-invasive testing platform where we need to go. And we still have a long ways to go for the drug development piece. But the picture is becoming clearer. And I couldn't be more excited to be a part of it than right now.
Stephen, that's a great last word. I want to thank surfers, Louise, Donna, particularly Stephen. I feel a lot better educated than I was an hour ago, and I know Donna and Louise do as well, and I believe the listeners will all feel that way after hearing this, or at least most of them. Please, listeners, we're working on the AASLD coverage design this week. If you've got anything you want to make sure we do or you'd like to hear us do, get back to us the next day or two. Send an email, put it in the discussion group. If you're on LinkedIn and you're connected to Hep Dynamics, send it over that way. If you send us our email address, I know more people are doing that every week. It's a good thing. And then beyond that, I just want to thank everybody we usually thank. Mike Wilson, our engineer, Buzzsprout, who publishes us, Eric Rounds, our social media master, Paula Teole, Ryan Segura, and you folks, our listeners and our subscribers, who help us as we continue to build the leading fatty liver podcast in the world and are starting to teach us more and more every week about how to do it better. We'll be back next week. My guess is we're going to try something slightly different with a special guest and a different set of issues. If it won't happen next week, it'll happen in 2 weeks. But we've got lots of good things coming up, and that's even before we get to AASLD. If you're in the States, by the time you hear this, you will have already experienced the first presidential debate. Try to stay sane amidst the crazy political season we're all living in. And wherever you are in the world, stay safe, surf on. See you on the podcast. Bye-bye now.